Hi everyone, and welcome to our final chapter in A&P 1 over sense organs. So we have two sets. We have chemical senses and we have other senses. Chemical senses are taste and smell. We also call these gustation and olfaction. And then our other senses are hearing, equilibrium, and vision. So taste or gustation. So we're going to talk a lot more about this in Anatomy and Physiology 2, specifically the lecture over digestion, but it's important to introduce here because we need to talk about physiological process of tasting. So the tongue has a bunch of tiny bumps all over it. These are called papilla. Papilla literally just means bump, okay? We have some tonsils in the back of the throat, and we have a lingual tonsil at the base of the tongue, but we won't worry about tonsils this semester. We'll talk about tonsils next semester. Well, let's look at the diagram. Do you see the upside down V shape toward the back of the tongue right here? So in this image from the textbook, it's called vallate papillae. We call them circumvallate papillae. So circumvallate papillae, inverted V near the back of the tongue. Circumvallate V. Fungiform papillae are small bumps on the entire surface of the tongue. If we were to zoom all the way into a fungiform papilla, they would look like little mushrooms. So these are these individual bumps all over the tongue. We also have foliate papilla on the sides. So these guys right here, those are foliate. You can think of them as half of a leaf shape, foliage, foliate papillae. You rake your leaves to the side. Foliate papillae are on the side of the tongue. It's a way to remember that. And then our last kind are filiform papillae or papilla. Filiform fill in the rest of the tongue. So fungiform are these individual bumps all over the tongue. Filiform are these tinier, smaller ones all filling in the rest of the tongue. They fill in all of the empty spaces of the tongue. They're the fill-in, the filiform papilla. Types of taste that we have are sweet, sour, salty, bitter, and umami. Umami is that beefy taste. Notice that spicy is not on here. Spicy is actually a pain receptor, not a taste bud. Okay. So all these little papilla that we talked about, these bumps on the tongue, people will call them taste buds, but they are not technically taste buds. Okay. Papilla just mean bump. If we zoom into one papilla, so this is a foliate papilla. This is one. You guys see these things on the sides right here? All of those are taste buds. So you have taste buds embedded in the sides of the little papilla or bumps. The taste bud openings are all down the sides. So each taste bud has a taste pore, and that's our opening right there, all down the sides of the tongue. So not the tongue, I'm sorry, all down the sides of the individual papilla. So one papilla has multiple taste buds. So this is a bump. The taste buds are embedded inside the single bump. So we've got multiple taste buds per papilla. Okay. And then lastly, cranial nerve 7 goes to the anterior 2/3 of the tongue, and then cranial nerve number 9 goes to the posterior. So be able to differentiate between the types of papilla, the types of taste, and where the cranial nerves go. And then where the taste buds actually are within a papilla. And yeah, that's it for taste. Let's talk about smell. We've talked about olfaction and smell several times throughout the semester, and we're not done quite yet, but we are just going to touch on it here because we have talked about it so much. We're not going to spend a significant portion of this lecture on olfaction. But first, let's talk about the cribriform plate. So that's right here, the base of the skull. cribriform plate. We've got the olfactory bulb right there with the olfactory nerves coming off of it. And then we have a membrane right here at the nerve endings. When you breathe something in, when you're breathing and a smell wafts up there, those chemicals get picked up by the nerve endings right there, and then they get carried along the olfactory nerve to the brain, to the sensory cortex. Remember the postcentral gyrus, posterior to the central gyrus or central sulcus, which separates precentral gyrus and postcentral gyrus. Anyways, that olfactory nerve is carrying that sensory information to the sensory cortex, which is the postcentral gyrus. A condition that can go with this is anosmia. So that is a complete lack of a sense of smell. Those folks can't smell, and we don't know why they can't smell, but it does exist. All right, now we're moving on to other senses. So we're moving away from chemical senses. So know that taste and smell are chemical senses, and the rest, vision, hearing, equilibrium, are all other senses. You will be asked. All right, anyways. We are going to spend some time on vision, hearing, and equilibrium because we really haven't talked about that at all. We have talked about smell a lot throughout the semester. And then taste is just a quick explanation of where the taste buds are and the different forms of papilla. But anyways, vision is a big one. Big safety sense for the body, especially evolutionarily, right? Let's talk about parts of the eye first. So sclera is the white outside layer of the eye. So if you're looking at this model, the sclera is right here. White outside layer, and notice no sclera right here. That's called the cornea. So when we get to the front of the eye, it's a clear layer that you can see through, and that is called the cornea. Rest of it's sclera as the outer layer. Clear part is cornea on the front of the eye. Then we have the vascular part, so that's the middle layer of the eye, which happens to be the iris and the pupil. So the iris is the colored part, and then the pupil is actually not a structured part. So if you're looking in a mirror, you see the black spot in the middle. That's your pupil, but the pupil is just a hole, and this is where you learn that the pupil is not a structured part. It is just a hole. That's it. And then lastly, the inner layer, or the inside towards the back of the eye, we have our retina and the optic nerve. So retina, and then optic disc that we'll talk about soon, and then the optic nerve. Okay, let's see. Is that all I want to say? Yep. Okay, moving on to fluids, we have aqueous humor, which is between the cornea and the iris. So the aqueous is going to be anterior. And we have our vitreous humor, which is between the cornea and the retina. So vitreous humor is posterior. And those are our fluids. So aqueous humor, anterior. Vitreous humor, posterior. Some accessory structures that help protect the eye. So we have eyebrows, eyelids, and eyelashes. All of these help the eye from getting scratched. Eyebrows are going to help prevent sweat from dripping into your eye. Eyelids, also called palpebra. Know that, you will be asked. They keep the eye moist. And blinking helps protect the eyes from foreign objects. So if you're hiking or walking or running or biking or doing anything outside, any sort of, physical activity where you're moving in a forward direction, your eyelids and your eyelashes are going to work together to prevent an insect from getting into your eye. So that's going to be good for you because that does not feel good. I've had it happen to me where my eyelashes and eyelids were not working together like they were supposed to prevent it. Wasn't good for the bug either. So these accessory structures are really important in preventing foreign material, foreign objects, and helping in protection of the eye. And remember, this pupil, that's just a hole, okay? It's just a hole, not a structured part. All right, cranial nerves to the eye muscles. Cranial nerve 3, medial rectus, superior rectus, inferior rectus, and inferior oblique muscles. We talked about this in muscles and cranial nerves lecture. We're just going to touch on this to remember it. Cranial nerve 4 goes to the superior oblique muscle, the little loops that you can look at an angle. And then cranial nerve number 6 is lateral rectus muscle. That's the muscle on the outside of your eye. And that's it. We're not going to spend time on this. Vision and photoreceptors. Rods and cones. Okay, so you've heard about rods and cones forever. Rods are going to be more sensitive to light, so they let you see in dim light or low light. That's what gives you the gray and fuzzy outlines. Basically, rods help you see in the dark. Cones are for color, so that should be easy to differentiate. Cones, color. Cones are better in bright light, and your color vision's going to be in shades of blue, red, green, and then the blending of those three. Cones are for color again. Rods are gray, but give you night vision. That's the best way to remember these. And then some people have stronger photoreceptors than others. They can have more sensitive rods, which makes them better at, seeing in the dark, essentially, or more sensitive cones, which let them differentiate between colors a little bit more, so they'll be able to see more color spectrum than you can, for example. More photoreceptors. Let's talk about the optic disc. So this is the optic disc in this picture. When you shine a light in somebody's eye, the yellow disc is the little bright spot that you see. And that's where the optic nerve leaves and the blood vessels enter. So that's also a little bit of a blind spot for folks right there at that part. So if the image you're looking at happens to reflect right in the spot where your optic disc is, you're not going to see that image. That is your blind spot. So the optic nerve right there is what picks up your visual information and carries it, through the nerve, crosses in the middle of your brain, and ends up in the visual cortex in the back of your brain. But if an image hits this right on the money, blind spot, you don't see it. So that's pretty interesting. Stereoscopic vision. All right, so this kind of explains how your eyes work together and the way in which they work together. So I'm going to read through it, and then I'm going to simplify it. So when the eyes converge on the fixation point F, more distant objects D are focused on the retina's medial to the fovea, which is your focal point, okay? So they're going to be more this way for distance. The brain interprets them as being farther away than the fixation point. Nearby objects N are focused lateral to the fovea and interpret it as being closer. So near objects are going to hit here. So basically, if F is where you're focusing, anything with a farther distance is going to focus more to the medial part of the eye, so more towards the center. And you're going to interpret that as far away. And then closer items, that direct angle is going to be more on the lateral part of the eye, so outside part. And you're going to interpret that as closer. And stereoscopic just refers to your eyes working together for this process. Again, visual pathway, I'm going to read through this and then I'm going to make it a little bit more simple. Blue and yellow indicate the receptive fields of the left and right eyes. Okay, so blue, yellow, and then green, you can see the overlap. Green indicates the overlap area and stereoscopic vision. Nerve fibers from the medial side of the right eye, so red, crosses to the left side of the brain, while fibers from the lateral side remain on the right side of the brain. Kind of, I'll explain that. The converse is true of the left eye. The right occipital lobe thus monitors the left side of the visual field, and the left occipital lobe monitors the right side. Okay, so let's simplify because this is true but not completely what's happening. So you can see the right eye, the left eye. Okay, you can see the red fibers coming off the right eye, blue fibers coming off the left eye. Some of these fibers actually go to both sides, right? So the red's not necessarily all going this way and the blue's not necessarily all going this way. You have some fibers that go that way and some that go this way. So they are actually going to both sides of the brain. The purpose of this is that your body has a backup system. So if something is wrong with one side of your brain, your body has these backups in place, so it has multiple fibers leading to both sides of the brain from each eye, so that if one part of your brain is damaged, your body has a backup in place to communicate and send vision signals from both eyes still. And it also helps with stereoscopic vision, of course, too, because you're getting information from both of your eyeballs still, even though that part might be damaged. Does that make sense? So that's why it's not necessarily just right to left and left to right, because you've got some red fibers that go right and some that go left. You got some blue fibers that go left and some that go right. So redundancy system, should there be brain damage on the left brain maybe, you're still getting visual information from both eyes to your visual cortex, to your occipital lobe. Okay, hearing. So outer ear is pinna and lobule. So earlobe, okay? Pinna, lobule. So here we have cartilage in the pinna and then fat in the lobule. Be careful with piercings. If you have a piercing in the cartilage and the person makes some kind of, not necessarily, the person might not be making any mistakes, but they might pierce the cartilage in just the right spot that the cartilage can crack and it can just flop over. So this isn't very often, okay? But this is a cautionary tale that it can happen. The more you know. Anyways, pinna is the main part of the outer ear, and then you have the lobule or the earlobe. So pinna and lobule, outer ear. Middle ear, ossicles. Okay, so ossicles, remember anything OSS is bone, osseous bone. So ossicles are your ear bones. Here we have the stapes, incus, and the malleus. So if we were to look at a cadaver, middle ear, here it is. Know these, okay? Know these. Know that the stapes is the stirrup, the incus is the anvil, and the malleus is the hammer, okay? Malleus, hammer, incus, anvil, stapes, stirrup. You will be asked. The inner ear is our labyrinth. Here we have semicircular canals. So those are these semicircular ducts or canals. They're just this system of tubing. And then we have this snail-like cochlea. So that's where our hearing and our balance comes into play. Semicircular ducts are for balance and equilibrium. The cochlea is for hearing, and then we'll talk about stereocilia and how that happens next. Okay. Let's talk about the methodology of hearing. How do we hear? So sound enters and vibrates the tympanic membrane or eardrum. Okay. The eardrum moves back and forth. That means it moves the malleus or the hammer. That in turn moves the incus or the anvil, which then moves the stirrup or the stapes. The stapes is attached to the oval window right here. And there's going to be fluid on the other side. So when the stapes moves, it actually makes a wave of fluid going this way. And it comes around and makes more waves this way towards a round window. So, oval window up here, movement from the stapes causes a wave, and that wave goes this way as well. Secondary tympanic membrane and round window. As the fluid pushes on that membrane, we have stereocilia. So stereo audio, cilia hair cells. Stereocilia are auditory hair cells. And these stereocilia bend. Okay, straight up here, bending here. Every time the cilia bend, that triggers a nerve below. By the way, loud sounds can cause big waves that break the cilia, that destroy the cilia, which leads to hearing loss. And our cilia, our stereocilia, they do not regenerate. Some animals do have auditory hair cell regeneration, but we do not. So hearing protection, earplugs, this is really extremely important in loud environments to prevent hearing loss, because once you lose these guys, you're not getting them back, which means you're not getting your hearing back. But anyway, that nerve down here is the vestibular cochlear nerve, or the auditory nerve, and then that information is carried to the auditory center of the brain, and that's how all of that works. Cochlear implants are always an option for folks that have experienced hearing loss, but if you are able to prevent breakage of your stereocilia, that's better. That is better to stave off hearing loss. The auditory tube of an infant and an adult, we're just going to touch on. We also call this a eustachian tube. So that's the drainage tube from your ear to your throat, essentially. It's the ear to the nasopharynx, kind of right above where the uvula is, and that dumps into the throat. You've heard of postnasal drip? That's that area. In infants, it's really short and horizontal. In adults, you're going to have a longer face. So that auditory tube is going to be a lot more vertical in shape. It's going to be a lot longer. Anyway, it allows for better drainage of the middle ear. With a more horizontal shape in infants, it's really important not to accidentally cause middle ear infections by anything that you're doing. So, for example, if you're laying a baby down flat to feed it, you can actually cause a middle ear infection. That milk can get stuck up here and just it's a moist area. It can cause anything to sit and fester and inflammation, and that's what causes a middle ear infection, right? So you have to prop that baby up, okay? You might not want to. You might think, oh no, but the baby's so content laying on its back, drinking this milk. Nah, prop that baby up. Don't let the milk go to the ear. Don't cause an ear infection. The milk can sit, it can fester, it can get inflamed, cause an ear infection. Baby's miserable, you're miserable. So just prop the baby up when you're feeding it, right? Again, the more you know. And then propping that baby up, again, it just allows for better drainage so it doesn't sit there and fester and cause issues. All right, so we're going to end on equilibrium and balance. So we've got the cochlear part of the nerve right here that goes to the cochlea, the snail shape. And then we have the vestibular part of the nerve that goes to the semicircular ducts or canals. So here we've got even more cilia and stereocilia. So let's zoom in. Boom. Notice these big purple cilia. Okay, so we've got a bunch of these little supporting cells. And we've got these big purple cilia. We call these kinocilium, the largest stereocilia with the tip embedded in an otolithic membrane of the semicircular canals. Anyway, so these big purple cilia are large stereocilia, and they're called kinocilia. The tips are covered in a gel-like substance with little rocks embedded in them called otoliths. So oto, ear rocks. When you tilt over, notice how the otoliths are leaning down and they are actually pushing the stereocilia over. So the bottoms of these cilia are connected to the nerve that then translates all of this as, oh, we're tilting over. We're tilting to the right. And that's the nerve that lets your brain know, hey, I'm tilting to the right now. When you sit back up, everything levels out and the cilia are back upright, and that signal's getting sent down the nerve to the brain, right? And we're sitting back up. So sometimes these otoliths can get stuck, okay? So even though you're upright, if you were just tilted over and the otoliths got stuck, your stereocilia are stuck bending to the right because your otoliths got stuck. So even though you're sitting upright, your stereocilia are still bent over. Your other ear is telling your brain, hey, I'm upright. But now this ear is telling your brain, hey, no, I'm actually tilted over. And now your brain doesn't know what to do. And that is what causes dizziness. So anytime somebody says, oh, I've got an inner ear problem, I'm so dizzy, they're talking about an inner ear condition called labyrinthitis. Okay, and that's it. That's it for this chapter.